I talk very fast, and so this, but it kind of like musically, it's like a rhythm. With things I think that everybody should know just by being a Homo sapien, I'll talk really fast. When I get to things that I think people are not going to know, especially in a mixed audience like this, I'll slow down. I strongly advise you to interrupt me, raise your hand and say, wait, like, what does this actually mean? Because I'm going to talk about a lot of biological stuff that may not make any sense unless, no matter how carefully I try and explain it. So let's get started. So HIV is the pandemic of the 20th and 21st century. It remains a giant pandemic. Where are we now? How do we get here? And where are we going? Because in the end of the day, for all diseases, you only have three options. You can prevent them, treat them, and cure them. HIV has swallowed all three objectives. And today we're going to talk mostly about prevention. So most statistics are about 2 years out of date, but things haven't gotten that much better in the last 2 years. This is the UNAIDS report that shows probably there's now about 40 million people living with HIV. Probably about 20 million people have died from HIV, and probably about 2 million new cases of HIV occur each year. So it remains a really, really big problem. The disease, when it all started, and I was here when it all started, and that's a whole other story. When it all started, there was kind of a belief that everyone should get tested all the time, and that, you know, the best way to prevent this was to avoid the stigma associated with being in some sort of special group. That really doesn't work very well. There are people with excess risk, and this slide shows you the excess risk. Sex workers, 13 times more likely to acquire HIV. People who inject drugs, PWID, 22 times, and so on and so forth. The United States, of course, unfortunately, we're very focused, and I'll get to it in a second, on gay men, especially gay men of color in the South. I guess I'll get there right now. So, a warning to myself. So the US government, in the last 3 years, and this has been a really, really complicated effort of HHS leaders and the NIH leaders and the CDC to kind of argue that because the tools have been developed that might allow us to dramatically affect HIV in the United States, that we should aim to get HIV new cases down from about 40 to 50,000 a year to about 8 or 9,000 a year in the next 10 years. And I'll get into the tools in a minute. And the reason it's believed that this is possible because there's such a concentration of HIV geographically and in terms of risk group. So geographically, 51% of all HIV is in the South. And then the US is divided into about 5,000 to 6,000 counties, and I guess the demographers know better the exact number of counties than I do, but there are counties that are hotspots for HIV. And so it is believed that by focusing the resources on the counties and the people who are at risk for HIV, that the epidemic can be greatly affected. Now this slide gets at this issue that I spoke about earlier, that if you looked at 40,000 new cases a year in the United States, and I don't have a pointer, probably 30,000 are among gay men, and 28,000 of those are going to be in gay African-American men. And so trying to avoid stigma is one part of it. Trying to focus your resources is the other part of it. And that reaching that happy medium is extremely difficult. And that's what the US is going to try and do. And that brings us to the meat of how do you do this, okay? So on this slide is everything we know how to do in HIV prevention, not all of which is available. First of all, there is an introducing or communicating and inspiring safer sex behavior. And my friend Audrey has spent her lifetime on this one topic and done a great job at it. And the good news is there are lots of things you can do, shown on this slide, that actually make a contribution. The bad news is it's very hard to measure the contribution. In other words, we have no idea what this pandemic would look like had we not done all the things we did with condoms and STD control and behavior change. But measuring the contribution on a global level is almost impossible because we don't do clinical trials that are kind of well organized to get just at what contribution you make. Condoms are required and always a disappointment because they clearly work. They stop STDs and HIV, but no one looks forward to putting their condom on. People don't say, oh, it's a long weekend, I can wear my condom all weekend. It's just not a device that's ever going to be popular. And so we pay lip service to this. We never really can achieve more than a certain amount of condom use. And it's very user and age dependent. We don't have a vaccine. I'll talk a little bit about vaccines in a few minutes, microbicides. The kind of mother lode of energy in the last, I guess, 10 years has been on the idea that when you treat people, you render them no longer contagious. So we're going to start there, but let me just show you how this is going to fit into the HIV program getting to zero. Here's a couple of articles about the HIV program getting to zero, ending the epidemic in the United States. And here's what is intended in the United States: to increase testing and diagnosis in the highest risk people, to improve the treatment cascade, and I'll show you a picture of the cascade in a few minutes, to use pre-exposure prophylaxis, antiretroviral drugs for people at the highest risk, getting a healthy person to take a pill, much as you would use such drugs for malaria. And then what's probably the most controversial thing and might be of the greatest interest to you guys would be cluster busting. So HIV in communities can occur in clusters, and it's possible for us to identify the nidus of the cluster and the rate of growth of the cluster and the people forming the cluster using social science tools, but also using the virus as a detection tool. So we can now see, in fact, we just published an article yesterday, we can identify with 98% accuracy directionality. We can say person A gave the virus to person B. However, there might always be people in the middle. But once you have these tools, you get into tremendous ethical and stigma problems. So we have all these tools, and these are the 4 pillars of HIV prevention in the United States. So what I intend to do is to show you how we came to agree on these pillars and to show you where we're going next to improve on these pillars. Can you all understand me at this rate of speed? Mm-hmm. Okay. So treatment is prevention, TASP as it's referred to. So this is a very simple idea. In the end of the day, everyone on the planet, if HIV is 80% a sexually transmitted disease, and you're in a relationship, there are 3 flavors of your relationship as it relates to HIV. If you and your partner are both HIV positive, you're called concordant positive. If you and your partner are both negative, which is most people on the planet, you're called concordant negative. But if you're positive and your partner is negative, you're called discordant, and serodiscordant or serodifferent, and then there's the risk that you'll transmit HIV to your partner. So most HIV transmission depends on being in these kinds of relationships. Now, when we first started messing with this, it was generally believed, this is now about 2000, that everyone who was HIV positive, that their partner was also HIV positive. And it was people in the School of Public Health, we worked a lot on understanding, as did other groups, that about half the time when you're positive, maybe a little bit more, your partner's negative. So you have an opportunity to interfere with HIV transmission. So the one way you would interfere was with couples counseling. You would say, don't have sex, don't have sex, don't have sex, or only have sex with, with this one partner. The other way you might interfere, which is what we got into, was to understand the magnitude and durability of benefit when you treat somebody who's HIV infected. This was not known until 2010, only 10 years ago. And basically, so we and others, I led a study called HPD-052 that demonstrated that when you treat an infected person, these were heterosexual couples, you render the person in the partnership that's negative at no risk. So there's 0 transmission probability. Subsequently, this was also studied in gay couples, and it was the same thing was found. And so this slide is my most expensive slide for reasons you'll see. Bruce Elbert's mouth moves. So you can see how his mouth moves. This took a graduate student a year to make this guy's mouth move. And so Bruce in this editorial said the results of galvanized blah, 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 blah. So this was a big deal. It was a science breakthrough of the year. And it led to a lot of stuff. And it led, mostly importantly to us, is a campaign led by an HIV positive guy named Bruce Richmond, the U=U campaign. And his argument was, most people who are infected live in a stigmatized, fearful world, and the rest of their lives they feel they're essentially vectors for disease. And if we can communicate to them that there's no risk of transmission, their lives will be lived better, healthier, and it will also increase testing, because therefore people won't be afraid. So Bruce launched this U=U campaign. Undetectable is uninfectious. And this has really become a global campaign that many people, and certainly the gay community, is very aware of. And in the end of the day, the success of U=U depends on what's called the treatment cascade. And it's for every disease, whether it's in psychiatry or medicine, there's a cascade. You have to make the diagnosis, you have to link the person to care, you have to retain the person in care. In our case, you have to prescribe antiviral treatment, and then you have to demonstrate that the antiviral treatment has suppressed the replication of HIV. And if you just look at these fall-offs, every step of the way, you can lose people in this cascade, compromising the power of treatment as prevention. On the one hand, it is possible when you have, if you had a one-dose treatment of a disease, you can wipe out the disease by treating everybody on the same day. That's essentially what China did with syphilis over in the 1950s. They had a huge syphilis epidemic, and they treated everyone with benzathine penicillin over a short period of time. They went from like 30% syphilis prevalence in Shanghai to 0% prevalence in the country by 1964. So with the appropriate tools, you can wipe everything out. But we have lifelong treatment, and this lifelong treatment then is subject to loss in the cascade all along. Yeah, please.
Yeah, I was just curious what the denominator is there. So 82% of what? You know, like 82% of
82% of all the people estimated in this particular time in this particular study to be HIV infected. There's about, in the United States, we call these the hard to reach people.
Right.
And a huge effort goes into finding 20% of people that we can't reach. So we can identify in the United States 800,000 or 1 million infected people. But the last 200,000 people who to a great extent must sustain the epidemic, they cannot be found. Then once the chronically infected person infects somebody else, that person that's newly infected has acute HIV infection. Acute HIV infection is the highest level of virus you'll ever have. So the person with acute HIV infection, if they're sexually active, can build a cluster. Because then acute goes to acute goes to acute. And that's this whole issue of clusters, phylogeny, and stigma. All right, now the next thing, so this popularity of treatment as prevention, TASP, led to U equals U. It also led to population level intervention. The World Health, the UNAIDS and World Health Organization started a campaign 90-90-90. Find 90% of the people on the planet, treat 90% of the people that you find, and suppress viremia at 90%. And their modeling suggested that 90-90-90 would wipe out the epidemic. But then a bunch of different investigators did big studies. And these are the big studies, all of which are published in the New England Journal, ANRS, South Africa, Botswana, Search, and HPD-071. By disclaimer, I'm the leader of the network called the HIV Prevention Trials Network. So my loyalty lies to that network in all results. Somebody else might have done a better study, but that doesn't get mentioned. Only the studies that the narcissistic approach to science. So HPD-071 PopArt, population ART, that was led by investigators at the London School of Hygiene, Richard Hayes and others. And that was 2 million people receiving ART, community randomized study. And all of these studies demonstrate a benefit at different levels of treating a population. But none of them get much greater than 30 or 40%. So it's not bad to have a 40% reduction in new cases of HIV, but it's certainly disappointing or insufficient. So now, even this, looking towards 2020, we have a very large group at Oxford that's trying to understand why we only see 30% benefit. And these are the explanations why population antiviral therapy is limited. But this would be true for any study any of you would do. Magnitude of coverage. If you've only treated 60% of the people and maybe the wrong 60%, you can't get the maximal benefit. Maybe in the communities you're working in, some people have resistance to antiviral treatment. So you're giving treatment, but it's not sufficiently suppressive. What about the untreated people? I've already mentioned to you acute HIV infection. They represent a huge force in spreading HIV. What about in-migration? We're treating a community, but what if a bunch of HIV positive people decide to move into that community, they're untreated, and they infect other people in that community? That has been the subject of a huge body of work by investigators working on the virus with kind of surprising results. Because we use molecular phylogeny, we looked at the sequence of the virus, we can see who's infecting who. And the results have been very surprising, and I don't know if they're all published, but if there's time, I'll come back to that.
Mike, can I?
Yeah.
There's adherence here, because you can cover.
It's the intuitive is the cascade, coverage and the cascade. So Peggy's pointing out it's not just the coverage, it's obeying the cascade. If you just diagnose, but you never link to care and so on and so forth. Thank you for that comment.
Yeah, I was actually going to say adherence to the medication, because there's side effects that people stop taking it or don't take it every day. That it's there's a behavioral side.
I completely agree. So intuitive to all this is the notion that you use treatment as prevention. The cascade at every level has a problem. Stigma for testing, side effects for viral suppression, resistance for poor drug taking, and so on and so forth. Of course, intuitive, I agree. Other comments?
Did the studies that you were just talking about, do you know, did they measure they got to the 1990 or how far?
Yes, several of them got, some of them got to close to 1990. Palmar got close to 1990, but it just shows that that 90, if the 10 or more % that's left behind remain sexually active and able to infect somebody else, they can rapidly build a cluster.
I just want to be clear on the study design here. Everyone in these communities gets treated, is that correct?
So each one has a slightly different study design, 4 studies, right? They're all not the same, all published now. The pop art study design is community randomized. So you have, it was supposed to be 3 different interventions. It ended up being 2 interventions. The standard of care, and then everybody gets treated. Like knock on your door, test everybody in the household, everybody positive goes on treatment, and viral suppression being measured and a lot of other stuff being measured. But every design was a little bit different, so I don't want to homogenize all the designs. Okay, so in the tools that are going to be used in our prevention strategies, treatment as prevention is not going away. Because the other point of this is people get to live a normal lifespan by taking their pills. So being identified and treated means you live 80 years instead of 30 years. And so there could be almost nothing more important for personal benefit than being identified. So treatment as prevention is now axiomatic. It's not going to go away, but it's not going to end the AIDS epidemic. So what else might we do? So the US government and some other groups love the idea of pre-exposure prophylaxis, an alternative tool. So in 2012, one of the combinations we use for HIV intervention, Truvada, which is a combination of FTC and 3TC, 2 different drugs that work very actively against HIV, that combination was approved for daily, in the United States, for daily usage to prevent HIV. So 1 pill a day for a man or a woman would prevent HIV. And subsequent studies suggest that, in fact, for men in particular, if they take that pill every day, there is like probably greater than 90% prevention probability by taking that pill every day. Much less well studied in women, but the assumption is it probably works pretty well in women, but probably not quite as well as it works in men. Angela Kashuba, who's now the Dean of the School of Pharmacy, who's one of our big partners, she and her colleagues demonstrated the pharmacology is quite different in men than it is in women. Success of this intervention depends on adherence. Who's the adherence? The adherence? Adherence. The success depends on if you take the pill. If you're not taking the pill, you are not protected. One of the questions that has arisen that we won't have time to go into is how often do you have to take the pill? For women, they probably have to take the pill every day. We say it's unforgiving for women. For men, they can probably take the pill in a varied schedule, a schedule called 2-1-1, 2 days, 2 pills before, 1 pill the day of, 1 pill the day after, and that's probably protective. So these different pharmacologies affect how forgiving this medication might be.
Sorry, Mike.
Don't be sorry. If you interrupt me, it's better.
I'm a pain.
I get bored myself.
Yeah.
I'll fall asleep.
So with PrEP now, people are less likely to use condoms. And so we know that if you have genital herpes or certain STIs, that you're at higher risk of HIV. Does this prevent that additional risk?
Yeah. Okay. I get you. Okay. I'm gonna. So attributable. So what she's saying is entirely correct. If you get rid of ART and you start looking at ways to enhance or amplify the acquisition or the transmission of HIV, there's nothing better to enhance it than an STD, whether it's an ulcer STD or an inflammatory STD. The person who's untreated with the STD gets much more HIV in their genital secretions and transmits more readily. The person who is HIV negative and is at risk for HIV then has an ulcer, they're usually more likely to acquire HIV. So now we go to PrEP. And what happens with PrEP, not surprisingly, and this is pretty well accepted now, but grudgingly in some ways, pretty well accepted that and people taking PrEP think, you know, I'm not so afraid of STDs. I'm done with condoms. Condoms are not that much fun, as we've already talked about. I'm throwing away my condom. So STD prevalence rises in places where PrEP is used aggressively. But then you've asked a really critical question. can an STD that's acquired because I'm not using a condom overwhelm PrEP? And the answer is no. So if you're on PrEP and you get an STD, a rectal gonorrhea, a rectal chlamydia, and you're having anal intercourse, PrEP still works as long as you're taking the pill. That's a really critical question that's been addressed. All right, now PrEP use has increased in the United States. So now the question is, we got an epidemic with 38 million, 40 million people. There's only 600,000 or 800,000 people on the whole planet using PrEP. More than half are in the United States. And the ones using PrEP in the United States are not the ones at greatest risk for getting HIV. So this is just the growth of PrEP. But on the next slide, let me see. On the next, I don't have a slide to show you this, but I can tell you that most people using PrEP in the United States are more like worried well in communities where untreated people are less common. Why is it important about untreated people? Because if you've treated people in the community enough, then the PrEP becomes superfluous because the treated people can't infect you, right? It's the same as the argument about what do you do about a couple. Index cases reliably taking their ART. Does the negative partner need to use PrEP? That's somewhat debatable. We sometimes give PrEP for a few months, but then after a while we stop the PrEP as long as the index case is suppressed. Does that make sense? So PrEP use has been ill-directed, that's the best way of putting it, and it's certainly not directed properly on the planet. Now, one of the problems is what we call now PrEP persistence. It's a variation on adherence, and if you start a man in the United States on PrEP, they'll take it for a month or two, and then as shown in the slide, they're just going to stop the stuff for whatever reason. If you try this in Africa, which we've done a lot, to give young women at the highest risk, 7-8% risk of getting HIV, you give them access to PrEP. You call them and phone them and text them and say, take your pills, take your pills, take your pills. Their curves look just like this. So the good news is we've got these pills that can prevent HIV. The bad news is people don't want to take them. Now, why do they not want to take these pills? That's almost a mystery. They don't make people that sick. There's stigma associated with having a bunch of pills in your pocket, so therefore they're put in these little magical, quiet packages. I mean, there's a lot of effort to make it less stigmatized and make it more desirable. But to date, no one has succeeded well in getting much more than half the people at risk prescribe the drugs to take the drugs reliably, and the attrition over time is very, very great. So what do we do? What do we do? So, and this is, where's my structural intervention friend? Okay, you all would think of a structural intervention, right? We could do directly observed therapy, right? That would be something, but we're gonna, for all the millions of people at risk, we can't do that. Does anybody else have another idea of what we might do with PrEP? Because people won't take their pills, but we want them not to get HIV.
Before that is answered, what do we know?
Do you have an answer?
What do we know about the social science research about this?
But why?
Yeah,
way beyond what I would be willing to discuss, like off the top of my. I mean, there's a huge. Audrey knows a lot about this, and there's a huge.
What's affecting the
Why do
cascade?
women not? What do we know about why women don't take their PrEP?
Oh gosh, I mean, there's a lot of qualitative research.
It's a huge literature.
I mean, but all the things you would expect: violence, poverty, chaotic lives, stigma because it's ART. People think you have HIV. The list goes on, but those are the big ones.
It's always been, and no matter how, the investigators are always kind of arrogant when they start and saying, I've now got the magical solution to this. And they start out, and then we measure drug in blood, and then we get the results, and there's no drug in their blood. And we have a study coming out called HBN-082, which the investigators felt, led by Connie Callum, felt like we're going to do this so well, they're going to take their pills, but none of them look that different.
I was just going to add that I've seen some studies with Wendy that the sex workers in particular have been told that they can't drink and take their drugs, and so that they go off on the weekends and don't want to take it on the weekends, and that sort of. And I don't know whether that's true, and I don't know. My other question was whether they're.
It's not true that they can't drink.
Yeah, I'm just wondering whether there's also studies going on that you would know about whether they're adjusting the medications so that they have different recommendations.
Yeah, there's no reason you can't drink, so that's just
But that's what they're being told.
Yeah. So what are we going to do? You volunteer what we're going to do next. We have to have a structural intervention.
You need a pill that they would take once a year or once a month.
Okay. So we're not going to have we're going to do something. We're going to kind of take away volition, right? And so there is a once. The answer is volition has to go out the window. So there is a once a month pill, and that's called islatravir, and that is just now being investigated. However, it's still a pill. And so maybe people won't take it once a month. So let's do something different. Let's involve CVS and Walgreens, okay? And so then the solution is, let's do something incredibly long-acting that's infused or injected. Let's give a shot or an implant, like a birth control implant, and just change the whole dialogue.
So what are the people who won't take their pills doing about birth control? Are they all on
Good question.
IUDs?
No, good question. So let me address the long-acting and the birth control all concomitantly. And so other questions before I keep going, and I'll put it all into a package. Okay, so having said that the daily pills are and it's funny because my friends at the NIH, who you see on television all the time, like when we approach them once in a while about like, oh, let's do some daily pill stuff, they just like vomit. They say we're not supporting any daily. We just had a conversation like this the other day about daily pills, and they said we're not supporting anything with daily pills. On the other hand, from a clinical trial point of view, because the daily pills work, they're in the background of every clinical trial we do. We have to get a standard of care that involves daily pills, but now it involves an assumption that's painful. They're not going to take their daily pills. So therefore, you have to weigh what's going on with the actual usage of the daily pill in designing a trial, which is very complicated. I won't go into that now. So long-acting topical agents, I'll tell you a little bit about vaginal rings. Long-acting oral agent, I told you about islatravir, which is called Merck's drug MK-8591. Injectable agents, there's a drug called cabotegravir that lasts at least 8 weeks and maybe longer. Implants that can last a year, that would be combined contraception and antiretroviral. Long-acting patches that are in development where you, like a smoking cessation patch that'll last 6 months, that can have birth control and antivirals. And then cell gel, that's the little UNC thing that was in the newspapers here, where Victor Garcia and his colleagues take, and the School of Pharmacy people take a liquid, inject it into your skin, and it forms a lump that then dissolves over 6 months or a year. And so they can protect a mouse, but you have a lump in your skin, but you might have a lump. Well, Peggy doesn't want any lumps. But most women at great risk for HIV or men might accept a lump that was like not an ugly lump. Anyway, that's called the cell gel, and Gates say for the UNC to pursue the cell gel activity. So all of these things are going on, and they're all designed to get away from daily pills.
Can I just get a point of clarification? Sorry, for the ones that are mixing with contraception, are they doing that for treatment as prevention as well as for PrEP version? I mean, are they just
currently, well, theoretically, Merck who knows how to make Nexplanon,
right,
and it has the injector.
Do it for both.
Theoretically, the problem is you need multiple drugs for treatment, not one drug.
Right.
So once you need multiple drugs, the real estate in the device gets too small.
So it's just prevention.
It's mostly prevention. Now, okay, so these rings, dapivirine rings. The good news is dapivirine is an antiviral drug put into a ring. The rings were given to women. Women use them at a certain level, and in these studies of dapivirine, they got a certain amount of protection. The problem is that it turns out that women don't like having a ring in the vagina during intercourse, and this dapivirine is not metabolized on anything stable. So a lot of the women took the dapivirine ring out when they're having intercourse, but then there's no protection, and not surprisingly, a lot of women acquired HIV. Nevertheless, the inventors of this, the Partnership for Microbicide, they're seeking approval from the European agency that covers Africa as well, basically, and eventually from the United States. And the approval for this has been tied up for more than 2 years, whether they're ever going to approve this. And it's very complicated. On the one hand, you're approving something that offers modest benefit. On the other hand, you're approving something that offers modest benefit. So to the extent that it might inspire more sex, that could be a problem. No benefit was seen in women under 20. And that's not clear whether that's a biological phenomenon or an adherence phenomenon. So there's a lot of consternation in this field about what's going to happen with these rings, and is there any future for the rings? Now, again, we're driven to a great extent by the NIH. The NIH supported a whole network of topical agents called the Microbicide Network, which they killed. So there is no Microbicide Network after this year, because NIH is much more concerned about interventions that are more structural and long-acting. Did you have another question?
We have contraceptive rings that women don't take out, so I'm sure what's the
only
difference?
because, right, there's a contraceptive ring which has never proven popular. Only 4% of the market, even in the United States, are those rings. Now, you've raised a really good point, though. Multi-purpose technologies. The way rings become attractive is combining an antiviral and a contraception in a ring that women might find attractive. The way pills would work for women would be, and this is being developed right now, is oral contraception and Truvada in one big pill. Not so big you can't swallow it. So the POP Council is making that pill, and we're anticipating testing that pill next year. So contraception plus antivirals is a very attractive hook for women.
I'm just surprised they're taking it out in this case where we don't take it out for.
Well, I don't know. Are you volunteering using that?
No, I'm just saying,
Who's
that
the
my
we?
understanding
Who's
is that women are, I mean,
I don't know enough about the efficacy. I mean,
They do take it out.
Yeah.
It's not very
I
widely
also
used.
the other problem with taking it out for the contraceptive ring is the benefit is longitudinal. Taking it out for a day probably doesn't compromise the benefit. It changes the mucus and the consistency of, what's the right way to put this? There's no way I can say this right. So the way the ring works allows more forgiveness than Depivirine ring will allow. Did you? Okay.
Does anyone do trials where you let women within a community pick which of these various options they want so that you sort of take advantage of some selection feature where they're making the choice?
The ECHO trial, did they have any volition in the ECHO trial? Do you remember?
There have been trials and where people have, they've given options.
ECHO trial, I think, had some, and I should know this.
They assigned
Pardon?
them, they randomized, but they allowed them.
Yeah,
I think the ECHO trial had
Yeah,
some,
ECHO, I think they got
thank you.
changed.
Yeah. All right, that's the ring. Let's go on to something else. Okay, HPT and OA3. So there's two studies, HPT and OA3 and HPT and OA4. These are giant studies. These are studies of an injectable agent called cabotegravir. Now, cabotegravir plus another drug called rilpivirine, that is a combination that is up for approval to inject for treatment, a two-drug treatment combination every 8 weeks. Right now it's 4 weeks. In Europe, it's every 8 weeks. It'll be every 8 weeks. And the notion is that some people who are getting pills with infection don't want pills, and that they would prefer to get a shot. And in fact, when you ask the users about the shots, there's tremendous interest in getting a shot. You can understand that a very desirable group for that would be discordant couples, where the infected person would be enrolled as a couple, and then you'd know that they were suppressed because they got a shot every 8 weeks. You would understand that there's negotiations to deliver these drugs at the pharmacies and not in doctors' offices. So you would go, those little kind of booths at CVS and Walgreens, you would go there to get your shot. You would never go to an HIV clinic again, because we don't see people with HIV but once a year. So they would go to Walgreens to get their shots. So this is going to happen, the treatment part of it. Why is it slowed down? It was up for approval in December of this year. There was a manufacturing issue. And so the FDA said, we won't approve this combination until you resolve a manufacturing question. So we anticipate that this two-drug injectable combination for HIV will be available in the first quarter of this next 3 months. Okay. In that combination is the drug cabotegravir. That's a drug that is an integrase inhibitor, which is our best class of drugs, and it doesn't have a long half-life. It's milled into a suspension called the nanosuspension, and it's shot into the muscle, and it's slowly absorbed from the muscle. That's good news and bad news, because it's so slowly absorbed, it's like plutonium. You know, like which we say, which has a longer half-life, plutonium or cabotegravir? Because especially for women who have somewhat different fat and muscle in their buttocks, women will retain some level of this drug for more than a year, maybe 15 months, 18 months. So once it's in, we can't get it out. Having said that, there's no way for us to dialyze it or get it out. Having said that, we have a study now, a men's study called HPT and OA3 and a women's study called HPT and OA4. It's a double-blinded randomized control trial of the pills, Truvada pills, versus an injection every 8 weeks. That study is really just about fully enrolled. There are 27 sites in the United States, so on and so forth. There's 4,500 people enrolled, and we're enrolling 500 more, so there's going to be 5,000 people in that study. Then for the women's study called HPT and OA4, we have 3,200 women, 33,000 are already enrolled. These are all enrolled in Africa. These are very high-risk study subjects. And we anticipate, now this is high danger, because every 6 months, we have no results. We meet with an oversight board of priests and lawyers and statisticians, and the oversight board knows what's going on. And every 6 months we hear, will the study continue or will be stopped? Will be stopped because of futility, because the drug doesn't work. Will be stopped because the drug actually works, and there's no ethical reason to continue. So our next meeting is in May, and so there's high drama and anxiety around these meetings. We anticipate that this drug cabotegravir will work It'll become an injectable prevention agent. It's a structural intervention, essentially, for women and men who have trouble taking pills. And if they come every 8 weeks, they'd probably be protected very, very well from infection. So that's where we are with a long-acting injectable agent. Now, I've skipped implants a little bit. Merck is making an implant. Vive, the company that makes cabotegravir, is making an implant. Gilead, the company that makes Truvada, is making an implant. All of these implants would be subcutaneous and last for months. And there's a very robust implant program going on. Merck is way ahead of everybody else because of Nexplanon. And Merck clearly would like to do birth control and antiviral at the same time in an implant. And they'll probably be successful eventually.
And are they going to be affordable? I mean, do they have a good price?
No, no one knows anything about any of the prices, but you can bet that they'll maximize the cost, right? I mean, they're not going to be like generous. They're going to say, how much can we charge to the benefit of our company? You know, but usually what they do is they charge Americans $10,000 and people living in Africa $10. So, you know, there's a huge pricing differential. And I assume that will transpire. But this is where we are with antiretrovirals. And I'm going to now go down a whole other road for 5 or 10 minutes. That's like a much more complicated road. Yes, sir.
In the same vein, I mean, when you were talking about the treatment cascade earlier, you see that the biggest drops off are getting hooked up to care and being maintained in care. So in the US, cost is a driving factor. People don't have insurance, people have poor insurance, and even if they're insured, they can't afford their medication. So
I think that well, there's truth to what you say, but most states, and there's blue-red differences, most states have programs with the pharmaceutical companies to make sure people can get treated. I don't think any state in the country doesn't have a program for people who can't afford their drug, because it would be insensible, right? Because you're just going to get more infections. So North Carolina has a very aggressive program where no one in North Carolina who needs treatment doesn't get treated because of lack of insurance. But I can't speak for every state in the country. I'm sure every blue state. But I think that the barrier, there's certainly no doubt that copays, if there's a copay, there can be 1,000 barriers that would be invisible to people like me, that would be very visible to somebody going to a clinic. So, I mean, it's got to be a wise statement.
But I mean, in the tuberculosis literature in the US, like that is just covered and you don't actually have to go to a doctor's office and pay your copay. You can just go to the health department. Is something like that?
It's not as, I don't think it's not probably as streamlined as the tuberculosis issue for a whole bunch of reasons. And that's a whole other, it's called ADAP, you know, the programs without people with enough money. And there was a waiting list for people to get their pills. And then once 052 came out and the prevention benefits became visible, those waiting lists disappeared, because you really have to treat people to subsume, to prescribe your epidemic. Okay, we're gonna go down a whole nother road. If we live through this road, that'll be a wonderful thing. Okay. All right. So in the end of the day, we cannot control an infectious disease without a cure, like the China syphilis, everybody gets benzoyl penicillin, or a vaccine. There's no disease that we've really prevented on this planet well without a vaccine. And you still need a lot of vaccine coverage. You see polio and smallpox really constrained. You see measles breaking out because of this whole vaccine fear. So we need an HIV vaccine. And there are two kinds of flavors of HIV vaccine development. One flavor is kind of historical. It's kind of the human's a black box. Let's make something, hope it does something good for the human, put it in their arm, see what happens. This is called active immunity. When you put something in somebody's arm, they have an immune response, and you pray that they're protected in some way that's magical, without knowing what the magic is. That's the historical way of doing this, and we've tried this now for 30 years, and there are two remaining. So there was a trial about 4 years ago called RV144. In that trial, they did this. The human was a black box. They got some modest protection, and then $100 million was spent to try and understand why there was modest protection. And that led to the belief that the modest protection could be recapitulated if we made a slightly better vaccine. And these two trials, P5 and the Janssen-Crucell trial, those are vaccines that are trying to recapitulate the historical RV144 vaccine. That's one way of making a vaccine. The other way of making a vaccine is kind of a more rigorous scientific approach, where you understand what you need to do, and you figure out how to make the human being do it, where you know in advance what the human being needs to do. That has led us down a road of using of doing passive immunity. Passive immunity means we're going to experiment with humans and infuse them with something that we think will prevent HIV that maybe we can make a vaccine with. So these are monoclonal antibodies, and in this case, they're going to be called broad neutralizing antibodies. Now, monoclonal antibodies are way in use in the United States. Who watches television between 6:30 and 7? And if you watch television, it's all about rheumatoid arthritis and psoriasis. There's no one on psoriasis that should not be receiving an injection. Those are monoclonal antibodies being used to prevent psoriasis. So we're going to talk about monoclonal antibodies briefly and HIV. So this is how this works on this slide. When somebody acquires HIV, that's the virus is acquired and the host wants to kill the virus. That's true of any infection you get. But for most infections you get, the virus is not dynamic. You got the virus, the host sees the virus, it starts making antibodies, it mobilizes cells, the antibodies to cells gang up on the virus, and in 3 or 4 days you're better. This is certainly what happens with influenza. In the influenza vaccine field, we give you a vaccine that prepares you to see influenza, if we're right about it, and you're already prepared to have some antibodies against influenza, so you'll be less sick if you get influenza. Now, HIV, this has not worked. So HIV, you've acquired HIV. When you acquire HIV, you get one virus. Like, it's not a bunch of viruses, you get one virus. That one virus then replicates. And as it replicates, it's got a special trick that's very undesirable to us. It lacks fidelity. So every time it replicates, it throws off another virus that didn't look like the first virus. Does that make sense? Lacking fidelity. Most viruses lack fidelity at some level. HIV is incredibly promiscuous. It lacks fidelity, it lacks fidelity, it lacks fidelity. So this first virus you got, the host attacks it, it makes antibodies. Those are the antibodies that we see that show you're HIV positive, right? But those antibodies you made are not protective because by the time the antibodies get to that virus, it's not there. It's another virus that's there. We think of this as an arms race. Does that make sense? Now, with flu, as a good example, the antibodies you make are actually effective. And so they kill flu. Flu's not so clever as to escape or it would look like HIV. Okay. So this goes on and on and on. And 90% of the time, people keep making antibodies. They don't. So once the next virus occurs, you have 1 billion B cells in your body that make antibodies. So once the next virus occurs, the host doesn't give up. It says, uh-oh, here's another antibody. Here's another virus. I'll make another antibody. The host keeps making antibodies, making antibodies, making antibodies until finally about 10% of the hosts start making antibodies that don't help the host at all, but they're so broad, they kill most of the viruses on the planet. Those are called broad neutralizing antibodies, or BNAbs. Now, of course, this only works for untreated people. If you treat somebody, they stop making new antibodies, right? So untreated people can go on and make BNAbs. And there are like now hundreds of BNAbs that have been described. And the BNAbs, work at different parts of the virus. The virus needs to use a particular receptor, the CD4 binding site, in order to bind the host. But the virus has other requirements. The antibodies that are developed, these BNAbs, work at different places on the virus, right? And so the goal is then that maybe there's a BNAb that would be just as good as the antivirals to prevent HIV. Or maybe there's a BNAb that will be important to treat HIV because it'll stop replication of HIV. Does that make sense? Okay, so here's a bunch of BNAbs. The CD4 binding site BNAbs, the V3 glycan BNAbs, the Emperor BNAbs, and the V2 glycan apex. Most of the BNAbs we're focused on are CD4 binding site BNAbs. So if you take an animal and you take this one antibody, VRC01. So this is a guy studied at the Vaccine Research Center at NIH. It's a human being. He's blood and blood and blood. Before treatment was available, they stored the cells. And only 15 years ago, 12 years ago, a technology developed where you could take one cell and from the one cell make a BNAb. So this guy, VRC01 guy, has a VRC01, VRC02, VRC03, all the way through VRC10 now. So looking at all the cells from better and better cells to make a better and better BNAb. What makes a good BNAb? 3 things. First of all, the BNAb needs to grow in a cell line so that you can grow a lot of the BNAb. Some BNAbs are so toxic to the cell that you can't grow it. Second, potency. How much BNAb does it take? If it takes a gallon of BNAb to kill a virus, it's not very good. If it takes a smidgen of BNAb, it's a really good BNAb. Third issue is breadth. How many viruses do you kill? Some BNAbs kill 60% of viruses, some kill 80%, some 95%.
If you're resigned to using passive immunity and you're targeting things that interact with the CD4 receptor. Why not just give people injections of recombinant CD4?
Okay, back to 1989. Good idea. Tried in 1989 with great fanfare and then went down the drain. There's in fact a whole book about it by a guy named John Cohen, C-O-H-E-N, called A Shot in the Dark, about your idea. So it's a great idea. You were not, you might not have been born at the time. You're a young guy. These experiments were done and they really went down the drain. But A Shot in the Dark, I don't know, you should buy it for the Bob Center.
Okay.
John Cohen is the journalist for Science Magazine. He's the most published guy in HIV because he writes for Science every week. Okay, so not a bad idea though. So we can't do active immunity against the CD4 binding site for a whole bunch of reasons that I've kind of dismissed, but it was tried really aggressively. So the passive immunity idea is a proof of concept. And the proof of concept is like, here's VRC01, let's give some VRC01 to an animal, let's take viruses that we know are sensitive to VRC01, put them into the animal, and the animal can't get infected. So the idea works, that's the first issue. But does the idea really work in humans? So why do we spend $200 million? So now, Larry Corey, who runs the vaccine network, and I run the prevention trials network, are the leaders of a study called the Antibody-Mediated Prevention Studies, AMP studies. The AMP studies are the proof that passive immunity will work to prevent HIV. So we have taken VRC01 and infused it, shockingly, like run an IV, into 7,000 people with 50,000 infusions, okay, at this point in time. This study is coming to an end now. It's unblinded in August. It'll be reported, the results will be reported in October. And we don't know the results. But so we've completed this study, basically. It's a study of men and in women with all these infusions, not to make a drug. We're not trying to make this as a drug. We're trying to prove that we can prevent infection, that the breadth and the potency of this antibody are sufficient to prevent infection. Now, there's another issue here that I don't want to forget about. All these passive immunity studies, they're good because maybe we'll make some antibodies that will actually prevent HIV, but they inform the vaccine field. So my colleague at Duke, Bart Haynes, is one of the best scientists probably in the world. He's figured out ways to make humans with a vaccine make these antibodies. So if we know an antibody works through passive immunity, and Bart can make a vaccine that forces the generation of that antibody, we will have a rational antibody generation plan. And it's very complicated how he's done this. I don't want to, remember the arms race I showed you, virus, host, virus? What he's done is mostly when you get a vaccine, you get 2 shots. The first shot and then a booster shot, for example. He's doing sequential vaccination with 5 different views of the virus, trying to recapitulate the exact antibodies that we're infusing into humans. So it's much more complicated. But he's doing it somewhat successfully.
So explain these antibodies a little bit more. Are they specific to HIV?
These antibodies?
Yeah.
Yes.
They are, so they're not going to touch any of the other viruses that are circulating?
No. These are directed solely against HIV. In our 50,000 infusions, they've proved, this particular VRC01 has proved incredibly safe. We've heard no one's died, which is like an amazing thing, knock on wood, from the study, and there's been very few serious side effects. So these are much safer than the things you see on television.
But if the virus is changing so quickly, how are you making an antibody that's specific enough?
Okay, good. So let's go back over this. Remember, this VRC01 antibody was discovered from the person who was untreated for 10 years, and that antibody already kills 80% of the viruses we find on the planet. It's preordained, right? We have an assay where we look at breadth. How broad is it? And we start with like the one antibody, there's one antibody here that the South Africans have made called CAP256. It is not very broad. It only kills 50% of the isolates, but it's very potent. You need just like a whiff of the stuff to kill. So that's why it's still in the game. Most of these antibodies kill 90% of the viruses we can find at a concentration we can achieve through passive immunity. We've done everything IV to this point, but now we're doing sub-Q. And then we're constrained by volume. How much can we inject into your stomach before you vomit, or feel, not in your stomach, in your soft tissue? So we're doing this. This study is coming to an end, the VRC01 study, the AMP study, AMP1 we'll call it. It's been done in the Americas and South Africa. As I said, about 7,000 volunteers. And then this was this proof of concept. So now in the fall, you'll start seeing in magazines or journals what's next. because now we'll start combining several antibodies together. We knew one antibody was never going to be enough. Now we're going to try and get 100% coverage by giving sub-Q 3 different antibodies. And there's a competition for which antibodies get to go forward. And this involves a place in India called the All India Serum Institute that's going to make the antibodies, and it's quite complicated. But this is where we are. And I don't want to go into the detail of which 3 antibodies. There's a competition, like a bake-off, for which antibodies get to be selected. So I want to come back to where I began. Okay, so this is everything we know how to do in HIV prevention. Obviously, there are many, many groups working on this. I've narcissistically summarized more the NIH view, but in general, this is what's going on the planet. What about the preventing HIV in the United States? The study we're doing is called HPTN 096 to assess an integrated approach that is put all the tools together, behavior and treatment and antivirals into one package to get 90% reduction in 10 years, especially in Black men in the southern United States. So you use HIV testing, PrEP uptake, ART uptake. You try and prevent HIV transmission, and you have this complicated kind of cognitive scheme where you're looking at all the variables that would affect the success of this study. So this will be a community randomized study of communities that get this more aggressive prevention strategy compared to communities that get the standard of care, trying to show you can get 50% better benefit in communities that get the strategy. These will all be communities in the South, Texas, Arkansas, South Carolina, North Carolina. Communities are being selected now and paired as demographers would do. And we anticipate this study will make a contribution to the plan to end the epidemic in the United States.
Mike, is Ada leading that one?
No, this study is being led by 3 people. Chris Byrer, who's an investigator at Hopkins, who's well known. Rob Remian, who's an investigator at Columbia, who runs the kind of behavioral institute in Columbia, and LaRon Nelson, who's an African American investigator, who I forget where he is. So that study is just, now we don't have the money for this yet. This is like a collaboration between the government, the NIH, the CDC, and us. And raising a couple $100 million is not that easy. So, but we believe this will happen. Yes, sir.
What is your assessment about the efforts?
About efficacy?
What's your assessment about the efforts searching for the cure? There's a joint venture at UNC between.
Yeah, sure. Okay, here.
What is that in this big picture?
Okay, I'm gonna then give an opinion of prevention, treatment, and cure. I'll give 3 opinions. Prevention. We're better off than doing nothing. There's no doubt we're better off than doing nothing. The epidemic would be much bigger if we did nothing, but we have not really succeeded in prevention at the level that would be appropriate to the magnitude of the problem. Treatment. That's probably the best thing that happened in the 20th century is simple, one pill a day, letting people live worldwide normal lifespan. It's quite amazing. There's really nothing else in medicine like this, where when I was here at UNC, we had 5,000 people with hemophilia on our ward in 1985. Almost all of them died. By 90, we had pills. And by 96 or 98, we had 1 pill a day. And it's really quite remarkable if you can get people on their 1 pill a day. So treatment, anything we do has to be better than treatment. It's really hard to do better than treatment. Cure. So cure, there are about two big flavors of cure. One flavor is gene therapy. That somehow you're going to inject into the human something that's going to confuse the virus so much that the virus can no longer infect cells. That's one strategy. That's a difficult strategy. The other strategy is called shock and kill. You try and reverse latency of HIV. When we treat people, all the virus in the blood is gone. When we stop the treatment, viruses that are hidden come back. If we could shock the hidden viruses to release all their virus, that's one way to start the cure process. Then once we shock the viruses, we have to kill every cell that released its viruses. Shock and kill. Next week in Nature, UNC investigators will publish 2 papers in the same issue of Nature. Those papers are designed to show a new drug discovered here. The new drug shocks the cells 1,000 times greater than anything else ever reported. It's been done in mice and rhesus macaques at the same time. So shock and kill is the big strategy on this campus. Gene therapy is a big strategy for the Gates Foundation and for NIH. And those two strategies will continue until we give up. We'll probably never give up. So I think that we will make progress. We won't cure people in my lifetime, but we'll probably create environments where people can get off their daily pills for long periods of time. We will put people into remission. Like the cancer field often doesn't cure people. It puts people into remission with 5-year follow-up. We'll probably succeed at remission in HIV. I doubt if we'll cure HIV, in my lifetime at least. Okay.
Can you end on something a little more?
Let's see. What's a happy note? You want me to end on a happy? Well, 2 papers in Nature in the same issue is crazy, wonderful. And you'll see a big newspaper story. Whether we cure HIV or not, the 2 papers in the same issue in Nature is like never going to happen. And I told the investigators are very driven. I said, try and be happy like for 3 minutes. Like, you're going to have 2 papers in Nature in this big press release. Never happen. Try and be happy like for 5 minutes. I'm sure they won't. Okay, thank you for listening. Thank you.